Thank you for joining us for our webinar, The Future Outlook of Food Allergy Treatments. I am Jennifer Gertz, Executive Director of Food Allergy Canada, and today our expert joining us is Dr. Cecilia Berin. Before we get started, I wanted to note a few items. First, this session is for informational purposes only and will not provide specific medical advice, recommendation, diagnosis, or treatment. Please talk to your doctor about any concerns or questions you may have regarding your health or the health of your child. All participants are muted so we can keep the audio clear for the webinar. Thank you to everyone who submitted questions upon registration. There are some common questions that we will aim to tackle in today's session. If you have additional questions throughout the session, please submit them in the question box and we'll try to get to as many as we can. And lastly, this webinar will be recorded and shared on foodallergycanada.ca afterwards. First, some background on this session. When we talk about treatment, it's not surprising that everyone wants to know what's available now for the treatment of food allergy. What is the treatment? What does it do? And how can I access it? While our speaker will touch briefly on the treatments that exist today, the primary focus of this session is about looking forward. What is the future outlook for therapies? For those of you whose primary interest is in what's available now, we do have several previously recorded webinars that you might be interested in, and we'll share where you can find those in our wrap-up slides. The discovery and development of treatment for disease is a long and careful process involving several important steps. It starts with understanding the disease, what's actually happening in the body, and specifically, in our case, what is the underlying process that makes our immune system see certain foods as harmful and triggers an allergic reaction. With this understanding, scientists then identify specific targets in that process that they aim to modify through treatment. So think about this session today as going back to biology class. We're going to learn about the science, how the understanding of the science behind food allergy has evolved, and the potential treatments that are under development that can then be evaluated for safety and effectiveness. Now I'd like to introduce our expert speaker. Dr. Cecilia Berin is a Bunning Professor for Food Allergy Research at Northwestern University Feinberg School of Medicine. She is a real expert, and she's had more than 20 years of dedicated effort against the space of food allergy, and for that, we are very appreciative of all her efforts to support our community. Her lab in specific looks at how the immune system relates to food allergy and tolerance. She focuses on understanding how the immune system affects how people respond to new treatments, exploring less understood food allergies that don't involve IgE, and finding out why some people are more likely to develop food allergy when they're young. I'd like to pass it over to you, Dr. Berin. Please proceed with your slides, and we're delighted to have you join us today.
Thank you for that introduction, Jennifer. I hope that you can all hear me. And so I'm going to be focusing on what's next on the horizon in food allergy therapy. I have some disclosures. I have no conflicts of interest related to this presentation, but I have done a limited amount of consulting work, which is listed here. So I've divided this presentation into three parts. First, I'm going to discuss the different types of allergen immunotherapy that are being used or are being developed for use to desensitize individuals to foods. Secondly, I'm going to talk about some new therapies that target specific parts of the immune system, so immunotherapies. And the third part, I'm going to be discussing therapies that use the microbiome, which is the bacteria that live in our gut. And before I get to the future, I want to talk about the current status of food allergy management. So when I first gave this talk 5 years ago, careful allergen avoidance was really the only widely accepted option available to prevent allergic reactions to foods. So oral immunotherapy for peanut, this is a commercial product available as an FDA-approved product in the United States, but oral immunotherapy in various forms has made its way into private practice quite widely. And finally, what was just approved this year in the States, and I believe is not as widely available in Canada yet, but should be, is the drug omalizumab. This is also known as Xolair, and this is an antibody that binds to IgE antibodies, which are the antibodies that cause allergy. And importantly, these are not cures. These are lifelong treatments that protect from allergic reactions. So just a little bit of background about oral immunotherapy. So this is the process of desensitizing to a food by starting with a very small amount and slowly, incrementally increasing the amount that's taken as a daily dose until what is called a maintenance dose is reached. So in the phase 3 study, this was a clinical trial that was published that really led the way to this being approved for use. They found that the majority of individuals, so more than 2/3, were able to tolerate 300 milligrams of peanut. So this is equivalent to about 1 peanut, and they started out at very low dose. So this protective effect does wear off if the daily ingestion is stopped, and that rate varies between different individuals. But it's based on clinical trials, it seems that having a lower specific IgE, so the IgE antibody levels to peanut, and being a younger age are factors that promote the development of long-lasting impact of oral immunotherapy. So we think this younger age at the start and lower IgE is important because we think that there's a maturation status of the immune system as the allergy is really developing. It's been referred to sort of as hardening of the immune system, that it becomes more difficult to reverse that process with time. So what are the pros and cons of oral immunotherapy? For most, it can increase the threshold to prevent accidental reactions and in that way really provide peace of mind. And if the goal is to incorporate food into the diet, which is usually a stronger interest for foods like milk, egg, and wheat, that can be achieved for many. So what are the downsides? For most, the actual treatment causes allergic reactions, particularly at the early escalation phase before reaching a maintenance dose. Now, for the most part, these are usually very mild, but they can be systemic. For some, the oral immunotherapy can't be tolerated. So about 10 to 20% will have persistent gastrointestinal problems, abdominal pain, nausea that doesn't go away and just makes the approach intolerable. And then for a few, so this is approximately 2 per 100, it can induce an allergic inflammation of the esophagus, which is the tube that joins the mouth to the stomach, can cause swallowing problems, and generally treatment has to be stopped in order for this to go away. So another con is that it only desensitizes to the food given. So for example, if an individual is taking peanut oral immunotherapy and they're also allergic to cashew and walnut, it's not going to treat their cashew or walnut allergy. So this balance of pros and cons basically means that this isn't a treatment everyone will want to pursue, but it is an important understanding, advance in our understanding of food allergy management. So where are we going from here? First, I think that there's a lot of interest in having more foods be available as a standardized low dose, particularly for those who are have very low threshold of reactivity to their foods. There's a lot of interest in developing alternative routes of exposure other than the oral route. And I'm going to talk a little bit about this approach, the use of grocery store foods for high threshold individuals. And the concept of threshold is really, I think a very interesting and impactful topic. So not everyone reacts at the same dose to peanut. So in clinical trials, for example, and in many food challenges done in the medical setting, these are done as increasing doses. So starting with a very low dose of the food and then increasing over time with giving the food every 20 to 30 minutes. And by monitoring for the first objective symptoms, that is when the oral challenge is stopped, and that is considered to be the threshold of reactivity. So we know from clinical studies over time that the threshold is pretty stable. There are some factors like exercise, taking um, non-steroidal anti-inflammatory medications, alcohol, things like that can alter the threshold somewhat, but in general, the threshold is something that is quite stable. So in most of the studies that were done for omalizumab and for oral immunotherapy, was really targeting these individuals with a very low threshold dose. And the goal was to try and get them to an intermediate dose so that they were then considered bite safe, or they were protected from accidental reactions due to cross-contamination. And what we know is that the majority of individuals are actually already in this intermediate range of threshold. And so these kind of approaches may not be of benefit to them. So when I was at Mount Sinai in New York, we wanted to know if children who were already at this high threshold could just use grocery store peanut, like peanut butter, to increase their threshold further. And to study this, we asked for volunteers with a history of peanut allergy to undergo this graded food challenge. I'm not going to show you results of this because they're still in the process of being analyzed, but we asked the individuals who were shown to have this high threshold of peanut allergy. They were divided into two groups to either continue avoiding peanut or to start including peanut into the diet at a dose lower than that would trigger symptoms. And then there are going to be two challenges coming up. We think that those who or we're hypothesizing that those who are eating the peanut will actually increase their threshold, and then we're going to test if they then avoid, how long lasting is that protection. So there's a lot of work going on in the science field to try and develop predictive lab tests. So some of these are lab tests that are already in use, like food-specific IgE or allergen-specific IgE, like an ARH2-specific IgE. But there's also additional lab tests, like epitope-specific IgE and other blood tests that are used in the research sphere that are being looked at to determine whether or not they could predict someone's threshold without having to do a food challenge, and to see whether or not that sub-threshold inclusion of food into the diet would be effective at increasing that threshold further. So in addition to oral immunotherapy, there's an interest in non-oral routes of immunotherapy. And one of those is sublingual immunotherapy. This is used either as drops that are held under the tongue, or there's a company that has developed a peanut sublingual tablet that is held under the tongue. So the dose range that's given, so I mentioned that the maintenance dose for oral immunotherapy was in the range of 300 milligrams. This is a hundredth of that. And it is a dose held under the tongue in order to be absorbed by the mouth mucosal system, which is thought to be very good at inducing tolerance. And there are a number of research groups that have been studying sublingual immunotherapy quite intensely with very good results, really quite getting to the rates that we see with oral immunotherapy and that level of protection. And like with oral immunotherapy, this is seems to do better in those with lower IgE to start with and at a younger age. And there's also still, trials in progress with epicutaneous immunotherapy. This is a patch that is applied to the skin daily, and there have been patches developed to peanut and to milk. Again, they're still in clinical trials. They use even tenfold less allergen than sublingual immunotherapy. And the key advantages of these approaches is that they tend to be safer than oral immunotherapy. They cause less reactions. The reactions are more localized to either directly on the skin or itching of the mouth rather than any sort of systemic side effects. But um, the downside is the amount of food tolerated at the end is proportional to that. So there's a lot of interest in trying to get some of these approaches, particularly sublingual immunotherapy, to increase its efficacy without increasing the side effects. Okay, so now is when we get back to biology class, and we're going to learn a little bit about the immunology of food allergy. And so this is really important for trying to understand what we can target to more specifically treat food allergy. So we know that individuals with food allergy, they have IgE antibodies. So those IgE antibodies recognize the food allergen, which are proteins within the foods. And in a sensitized individual, when they eat that food, intact proteins get absorbed across the gastrointestinal tract and bind to IgE that's on the surface of these allergic cells called mast cells or basophils. And these cells release mediators like histamine and platelet activating factor that are very important for driving reactions in distal sites like the lungs and the gastrointestinal tract and the skin. So we know that there's a lot of immunology that happens before you get the generation of IgE antibodies. So IgE antibodies are produced by a type of cell called the plasma cell. And plasma cells are a type of B cell, so they're differentiated from an immune cell called the B cell. So B cells are antibody producing cells, and they are instructed what to do by another cell type called the T cell. So T cells tell B cells what to do, and they tell them that through signals that include factors that are called cytokines. And the cytokine IL-4 shown here is the one that tells B cells to make IgE. So we have been doing a lot of research on individuals who have undergone allergen immunotherapy and have been helped by that, trying to understand what is the impact on the immune system that is responsible for that protection. So we know that mast cells and basophils after oral immunotherapy no longer can be activated through activation with allergen. We know that the level of IgE produced by these plasma cells drops over time. And we know if we study the T cells that they make different kinds of cytokines. And furthermore, not only do they stop making IgE, but the B cells change to a different type of antibody production. They make these antibodies known as IgG4. And these types of antibodies compete with IgE and prevent activation of these allergic cells. So based on that, what we know about what happens during oral immunotherapy and what we know about the immunology of food allergy, there are a number of approaches that are in current trials to make immunotherapy better and safer. So let's start with the safer. One of the things that has been done is the generation of a peptide vaccine. So a peptide basically it means that it's the whole protein, for example, peanut protein like AirH2. And instead of using the whole protein, which can activate mast cells and basophils and cause those reactions during therapy, this has been broken down into small peptide fragments. And these peptide fragments cannot activate those allergic cells, mast cells and basophils. But these little fragments are understood by the T cells. And in that way, they can reprogram the T cells that are orchestrating all of the downstream immune responses. So the idea is that this peptide vaccine would be safer and just as effective because it is changing the upstream T cells. There is also an approach in trials where peanut is being delivered in virus-like particles. And so This means that the immune system is seeing not just peanut, but peanut within something that it detects as potentially foreign and dangerous. And it's changing the way that that T cell instructs the B cells to make antibodies. So the idea is that it's going to push those B cells to make more helpful antibodies like IgG antibodies and less harmful antibodies like IgE. Okay, so that is some ways that immunotherapy, allergen immunotherapy, is being improved and tested, but there's also a lot of interest in whether or not we can target specific aspects of the immune response without having to give allergen. So there have been a number of different approaches, and first I'm going to talk about this here. So we know that mast cells and basophils are not activated after oral immunotherapy. They are suppressed. So can we do this without giving allergen? And this is an approach that has been tested. There's a group at Johns Hopkins University in Baltimore, and they have been testing this drug called a Bruton's tyrosine kinase, or BTK inhibitor. So what is a BTK inhibitor? This is a drug that has been developed for B-cell lymphomas, different kinds of cancers. And the reason that it works on B cells is that it inhibits signaling through a receptor on its surface. But it not only inhibits B cells, but it was also found to really profoundly inhibit mast cell and basophil activation. So the idea was, maybe we can give this drug and prevent allergic reactions, and they did that clinical trial. So in this clinical trial, they pre-treated individuals using 2 days of acalabrutinib, which is a type of BTK inhibitor, and then they looked at whether or not they were protected from a food challenge. So here they were doing a peanut food challenge. And what they found was that there was significant protection. So only 2 days of treatment with acalabrutinib really suppressed these reactions. So, where do we go from here? So the upside is, this is a medication that is very fast onset and also fast washout. So when you stop taking the drug, it stops working quite quickly. So what's the downside? There are other effects on cells of the immune system. So some of those side effects are because it's inhibiting molecules other than BTK. So these are called off-target effects that are affecting the body in different ways. Some of these can be severe. As they've been developing new drugs that are more and more specific, those side effects have been minimized. But there still is the problem that this signaling pathway, the BTK, is present in both B cells and mast cells and basophils. B cells are part of our normal immune response. They are, key for our protection from pathogens. So there would be a concern about taking this as a long-term medication, particularly in children. But if we can find a short-term use, for example, giving this drug together with something like an allergen immunotherapy that on its own might cause reactions, that might be a safer approach. Or for a short-term use, like protecting someone while they're traveling, may be an important use. A colleague of mine has used this to allow someone who had developed an allergy to their chemotherapy to get that chemotherapy, to get the treatment for their cancer without having allergic reactions. So I think that it does tell us that if we shut down mast cells and basophils, we can prevent anaphylaxis. And now we need to figure out how to shut down mast cells and basophils without affecting other immune cells in the body. So next, I'm going to focus on ways to inhibit this IgE antibodies that we know are reduced by oral immunotherapy. So this is now an approved drug, omalizumab or Xolair, so this is what's called a biologic. It is made from living cells. And different types of antibodies can be do different things. So, we know that the IgE antibodies that we, make in the context of allergy can cause allergic reactions, but IgG antibodies can protect us from things like flu or COVID. Um, and therapeutic antibodies are antibodies that have been engineered to be given to humans. And instead of doing things like binding to flu or COVID, they bind to other things we want to get rid of, like IgE. So anti-IgE antibodies, Xolair, have been around for a pretty long time and have been used to treat severe allergic asthma. So in a large study that was published earlier this year, the anti-IgE drug omalizumab was given to children and a few adults who had multi-food allergies. So they had to be allergic to peanuts and then at least two other foods. And the impact of treatment was looked at on after food challenge with the different foods. And what you can see here is that approximately 2/3 of the group that was treated with omalizumab was able to tolerate 600 milligrams of peanuts, so approximately 2 peanuts, without dose-limiting symptoms. And about half were able to tolerate a full peanut challenge, so that's 9 grams, without any symptoms. And then looking at the other foods that people were allergic to, there was some variability. Cashew seemed to be a little less effective, but egg and milk, also 2/3, were protected from these higher amounts of egg and milk. So this was very exciting, led to the FDA approval, and we know that omalizumab can be used in a couple of ways. So what was shown here was omalizumab as monotherapy. So the idea is that this is given, as a standalone therapy to give protection from multiple food allergies, and that's what the data supports. And the question is, and that's one of the questions that's going to be answered in ongoing studies, is whether or not foods can be introduced into the diet to eventually be able to wean off omalizumab. So that's a question that is currently being addressed. And the other question that is currently being addressed is whether giving this drug together with oral immunotherapy is going to be helpful for making oral immunotherapy safer, and then potentially allowing to wean off the drug. So what are the pros of this approach? A big reason why people are very excited about the drug is that this has multi-food treatment potential. It's not one food at a time, and we know that many people are allergic to multiple foods. And the con is that this doesn't stop IgE antibodies from being made. So this is a lifelong treatment with the potential exception of foods being introduced into the diet and allowing for desensitization in that way. Okay, so we know that IgE is central to food allergy. One way to inhibit allergic reactions is to block them, but it would be great if we could figure out how to prevent them from being made. So I mentioned that the cells that make antibodies are B cells, plasma cells, which are a type of B cell. They can make IgE antibodies that are allergenic, they cause allergic reactions, and then they can make protective antibodies like IgG and IgA. And the reason why B cells make those different antibodies, or the contexts why they make them, is that they are instructed by T cells to make these different antibodies based on factors called cytokines that they produce. So we know that IL-4 is a cytokine that drives IgE production, and then there are other cytokines that help with other types of antibodies. So we have another type of biologic drug called dupilumab that binds to the receptor for IL-4 that's shown here. And also IL-13 is a very similar cytokine, has very similar effects to IL-4, and this antibody blocks both of those molecules. So this has been a drug that has really revolutionized the treatment of many allergic diseases, including asthma, atopic dermatitis, chronic rhinosinusitis, and eosinophilic esophagitis. So these are, localized inflammation of tissues that are allergic in nature. So the question is, would they be effective in food allergy? So there are a number of ongoing studies, but then some that have already been completed. And these have been tested, dupilumab. So this antibody, I don't know if I mentioned the name, was dupilumab or Dupixent. And this was tested versus placebo as a monotherapy and together with oral immunotherapy. And these trials were done by the company that makes the drug, and the results were really not very promising. So there are other trials that are in progress, alone or with omalizumab to see whether or not there might be some, better application of this. But what they found was when they studied the um, allergen-specific IgE, they found that treatment with dupilumab reduced it. So it reduced it about 50%, and then it just plateaued. So it never really went away. And it's thought that there is this reservoir, that these plasma cells that make IgE, they they sit in the bone marrow, they're long-lived, and they no longer need those signals from the T cells. And so the question is, how can we get rid of those plasma cells? So, one of or the company that has developed dupilumab or Dupixent has another drug, and this is a drug that they have been using for a plasma cell cancer, and it basically wipes out the plasma cells. It uses an approach so that our own immune system kills off the plasma cells. So the idea is that that would get rid of all the plasma cells that are producing our allergen-specific IgE antibodies. And then the idea would be that IL-4 or dupilumab would be given so that memory B cells, which can repopulate the plasma cell population, they don't restore IgE producing plasma cells. They would be producing some other antibody isotype. So this is a study that is currently underway. It's a small study in adults. I think that it's very exciting it's going to tell us a lot about the biology of IgE and whether or not we can actually get rid of the production of allergen-specific IgE, which would be so exciting, not only for food allergy, but also for many other allergic diseases. The downside is that this is wiping out all plasma cells, so not just the bad ones, but also the good ones. So the idea is that repopulation from the memory pool is going to allow us to sort of reconstitute a normal immune system. But there is, I think, considerable concern about the side effects of this. But we are going to learn a lot from this clinical trial. Um, this is a bit of a sledgehammer approach, but I think if we can show that it works, then we can work to refine it and really identify what are the plasma cells that are only producing IgE, and can we get rid of just that subpopulation? Okay, we also have a number of immune targeting drugs that are there's sort of a network of immune cells that all talk to each other to drive the production of IgE or to make mast cells and basophils that cause the allergic reactions. And there are drugs available at many different points in this pathway. And many of these are currently in clinical trials. And I think what's going to be exciting about this is we're going to be able to see where exactly we can target to get the most effect with the least side effect. And one other, I think, a different type of approach that's being tested is to actually give IgG4 antibodies. So I mentioned that in response to oral immunotherapy, the normal response to oral immunotherapy is to make a lot of food-specific IgG4 that's thought to be protective. So here the idea was, can we just engineer these antibodies and give them and protect people from allergic reactions. So this is being done in the context of peanut allergy in phase 1 studies now. Okay, and for the last third of the talk, I'd like to focus on the gut microbiome, which I think is an exciting potential area for the treatment of food allergy or prevention of food allergy. So why are we interested in the microbiome in the context of food allergy? Well, one of the things that we know is there are certain lifestyle factors, such as growing up on a farm and being exposed to livestock in the first year of life, or exposure to certain pets, and this is primarily dogs that go outside the home. Um, these are some of the biggest protective factors against the development of allergic disease, not just food allergy, but multiple allergic diseases. And then there are other factors that have been found in sort of population-wide studies, showing an impact on risk of development of food allergy. And what these all have in common is that they affect our microbiome. They affect the bacteria that live in our gastrointestinal tract. So we know that the microbiome is different in food allergy. We know from studies being done on food allergic versus non-allergic children that the microbiome, and we measure the microbiome basically by looking at the bacteria that are present in stool samples that we get from kids who are part of the studies. And if we compare the composition of the microbiome, it is different in children with food allergy. Now we know, kids with food allergy probably do not have the same diet as kids without food allergy. Important foods like milk may be affecting the composition of the microbiome, but there are studies that have shown that the difference in the microbiome starts even before food allergy begins. So in that first year of life, before the development of food allergy, there's already detectable differences in the microbiome. And we also know that children who go on to outgrow their food allergy have a different microbiome than children who have a persistent food allergy, which really suggests that the composition of the microbiota may influence the course of food allergy. And this is strongly supported by basic science studies in this field. So as I mentioned, the microbial composition differs between infants who go on to not have food allergies compared to infants who go on to develop food allergies. And what labs have done is to test whether or not this difference in the microbiota is actually functional. And the way that they did that is they did what is called a fecal microbial transfer. They basically took stool from these infants and did a transfer of those extracts into mice that were missing their own microbiome. So these were germ-free mice, they don't have any bacteria, and they get the bacteria either from these non-allergic infants or these food allergic infants. And then they tried to make these mice allergic to foods, to peanut. And what they found was that the mice that had received bacteria from non-allergic infants were resistant to the development of food allergy, whereas those who had received bacteria from food allergic infants were susceptible to food allergy. So this really says that what's present in that gastrointestinal tract can influence susceptibility to food allergy. And importantly, in testing how that microbial composition differs, they were able to identify some bacteria that were missing in the food allergic individuals, add those back to these mice that were susceptible, and to then make those mice resistant. So on a mouse level, this is super exciting because this says that we can actually manipulate the bacteria in the gut to change susceptibility or resistance to food allergy. So can we do this in people? And there are a number of studies that are ongoing, a number of clinical trials, and these vary in their design, but in general, there is some sort of microbial therapeutic compared to placebo control, and looking at food allergy outcomes here, primarily looking at development of food allergy or resolution of food allergy when it's being used as a treatment. So there are a number of different approaches that are being tested. So the most crude of these is the fecal microbial transfer. So this is an approach that has been used for severe bacterial infection called C. difficile. It's life-saving treatment, basically involving the use of these standardized stool extracts from healthy donors that are packaged in capsules. There's a phase 1 study that has been done in peanut allergy, and it is being continued, some promising results. There are also studies using a range of probiotics. So I think that there's kind of two generations of probiotics. There's the first generation of probiotics, which have been known for many years. These are bacteria that are found in things like yogurt that are known to have an immunomodulatory effect. So there are a number of trials going on giving these probiotics together with oral immunotherapy to see whether or not that might improve the outcome. But something that I'm a little more excited about is the idea of using this next generation probiotics. This is things that were identified as being missing in those who were allergic. So these are derived from the human microbiome. They are associated with health, and these are being tested in clinical trials. We can also give prebiotics, which is basically giving the bacteria the food they need to thrive. So there are trials going on where fiber is being given in order to try and um, enhance the survival of bacteria that are associated with health. So a lot of very exciting outcomes or very exciting trials and studies are coming. So I want to just sort of flip back to the idea of the fact that it takes a lot of time to actually get therapies into the clinic. And this is in the range of 10 to 20 years, but what we have been discussing here is studies that are in human testing. So we know that in the past 5 years, there's been 2 new approaches, oral immunotherapy and omalizumab, that have been approved. And there are many approaches that are now in clinical development. So these range from phase 1, which is really assessing the safety of the drug, first in healthy individuals, and then testing the safety in those with food allergy. Phase 2 studies that are looking at both safety and signal of whether or not they work, and phase 3 studies that are really the larger studies that help to prove that these are, better than a placebo or standard of care, that are really testing the efficacy of those drugs. So there are too many of these studies to actually fit on the slide, so I put them here. And this is basically the studies that are ongoing that are testing different approaches to improve uh, immunotherapy for food allergy. And this ranges from studies targeting type 2 immunity, that IL-4 signaling, the virus-like particles, the fecal microbial transfers, and other sort of second generation probiotics. And then there are a number of interesting studies, including the BTK inhibitors. So basically, I wanted to emphasize that there is a lot in the pipeline, including new things that are really already at the phase 3 stage. And our goal is really to provide many options. So not just one size fits all, but really option that fits the goal of every patient and family. And of course, there's important work to be done to ensure that there is access to therapy. So with that, I'd like to thank you for your attention, and I would love to stop and take some questions. Okay,
All right. Okay,
great.
so that was terrific. Such a great overview of the science and really, provides the confidence that this is a, the idea around identifying these various different targets and then identifying therapies. So we did receive several questions regarding current treatments, okay, and who's best for them. And we recognize that many of these questions are best answered through a shared decision process between a clinician and the individual. So, but I'd like your perspective from the viewpoint of a scientist on when you look at the overall direction of what's being researched and what's available now and in the near term. How would you describe the individual who is best suited for these therapies?
Well, I think they are different, and omalizumab, for example, is really suited for those with multiple food allergies for whom oral immunotherapy is not an option, or um, who have difficulty avoiding those foods, or have severe um, comorbidities. Um, so I think that that the most exciting direction is really trying to get at the root of allergy, the common root of allergy. So rather than treating peanut allergy different from milk and egg and cashew allergy, to get at something that will address all of those, and to get at something that is um, you know upstream enough in the pathway that it
Right,
is going to be disease modifying. So, I'm very excited about the drugs that are really targeting the production of IgE, but
right,
I'm also very excited about the work that's being done in the context of threshold, and the idea that not all food allergy is the same, and
right,
that you know there are a substantial number of people who have this higher threshold, um, who uh, may really benefit from knowing what their threshold is, uh, being able to have lab tests available that can predict that threshold without having to do a food challenge, and to have options there. So, is avoidance best if you're already bite safe, or, you know, are there other options for immunotherapy that are available? So I think it's all about having many choices.
right, right. Okay, that is terrific. So let's talk a little bit about this piece around severity of food allergy, because you've highlighted, like, we don't know our individual thresholds, right? But it would be helpful to understand how the scientific community defines the severity of the food allergy and how that's considered in the development of treatment.
Yeah, severity is something that has typically been very difficult to study in sort of a controlled environment. There are severity grading scales, so from mild to fatal, I guess, is the most severe. And um, having a scale that takes into account number of organ systems and the severity of the symptoms at each organ system. So threshold is something that can be sort of easily measured in like a research setting, because people are undergoing these oral food challenges, these increased threshold. Severity is, we don't know how um, reproducible severity is. We know that threshold is reproducible, but severity of symptoms, we do not really know if
Right,
that is reproducible. And it is so it's so tied. Severity is tied to threshold and exposure levels. So it's a little bit muddy,
right,
uh, and so I think that um, um, you know, severity is, I think, a factor in who is going to get access so if someone has had a history of severe reactions,
Right.
they may be more likely to get access to things like omalizumab. Um, whereas um, other approaches like exposure to oral immunotherapy may be less of a feasible option. But I think, severity is a tough, it's such an important feature, but it's hard to study scientifically.
Right. Understandably. Yeah,
Yeah.
because you don't want to be using a system to create a severe reaction. So on when you say threshold is reproducible, you're meaning that when someone goes back in for an oral food challenge, they're likely to hit an objective reaction at that same dose level. Is that
Yeah.
what you mean by that? Okay.
Yes, and it's not
Okay.
it's not absolute. So we certainly know from the oral immunotherapy experience that things like exercise and illness, alcohol, these are things that can change that threshold. But in general, um, that so for example, if you look at the placebo group of a clinical trial, that generally their threshold does not change from baseline to post-treatment.
Go. Okay. Okay. That's helpful. So let's kind of go into a bit more of the future now. And one of the questions that we got was around stem cells. And might stem cells be used in the future to eliminate or treat food allergy?
Yeah, I think that that's an interesting idea. I think the use of stem cells, so uh, the use of stem cells is really going to be effective when there is, a cell type missing that we want to replace, or an aberrant cell type that we want to replace. And I think that, there are certain cells like regulatory T cells that are involved in sort of suppressing inappropriate immune reactivity that if we could, find a way um, to promote Treg growth from stem cells, that might be an interesting approach. The, ability to use cells that would um, drive more towards an IgG producing plasma cell rather than an IgE producing plasma cell would be useful. But I think, it's all about what those cells do after they get put into the body. And so there needs to be a really good understanding of how how they would be programmed to become the
To become
appropriate cells.
cells. Okay. Okay. Now, somewhat related, I guess, it's because it seems like there's several of the treatment possibilities that you presented today that exist in other disease states or indications first. So they've been shown to be effective in those diseases, and then they're considered for food allergy. Help us understand how that actually happens, and how the scientific community kind of collaborates to see those opportunities across disease states.
Yeah, I think that, the biology underlying food allergy and some of those other diseases is very similar. I think that, it's there's a good rationale for why a treatment would be effective in multiple diseases. I think that this will often get tested first in adults and as with sort of limited appetite for testing drugs first in children. And I think that that has been why many of these drugs are tested first in, adult asthma, adult atopic dermatitis, before getting to the sort of main food allergy community. Although, of course, we know that there is a large population of adults with food allergy who need options. But really, the clinical trials um, have in food allergy have focused primarily on children, and um, with some exceptions like those that I mentioned. Yeah.
Okay. So there is a question with regards to Zolar. In the other phases that are being done, is there an examination of omalizumab or Zolar to actually be able to be off that medication. That was part of one of the study arms, was it not?
Yes, yes.
Okay,
So that is being tested to see whether or not um, the introduction of foods, so it's sort of the third stage of that
right,
clinical trial, is to test whether or not introduction of foods can be done and whether omalizumab can be tapered.
removed.
Yeah, yeah.
Okay,
So
terrific.
there are mixed reports from its use about whether or not that will be effective, and of course it needs to be tested in a larger number, which is what this trial is doing.
Okay. I want to end with this last question. We've really appreciated your expertise, and I'd like to finish with this one with the time that we have available is, you know, you've been working in this space for so many years, and we so very much appreciate it. And from that vantage point of looking at the research that you do, but the research across the broader community, what are you most optimistic about?
I am most optimistic about the idea that we may be able to stop IgE from being made. I think the cells that make IgE are really fascinating. They behave very differently than cells that make other IgG antibodies. So the basic sort of immunology of B cells sometimes doesn't apply to these IgE producing cells. But there's a lot of information. It's coming very quickly now. A lot of um, research groups are using um, technologies that were not available 10 years ago to study these cells and to look at their contribution to production of IgE, where they're living, what they're dependent on for their survival, all sorts of things that I think are going to help us target those cells in a way that is more specific than just getting rid of plasma cells.
Okay. Well, listen, Dr. Berin, thank you so much. We do appreciate your expertise and you sharing that with our audience today. So I'm going to turn off, we're going to turn off our cameras. I've just got a few slides that I'm going to wrap up the session with, but thank you so much. All right. So let's just go through a few things around some of the research studies that are underway today, because we would like to share with you which ones are seeking Canadian participants. So look on our participate in research page at foodallergycanada.ca/research. You're going to find the trade study. This is a study that's seeking children age 1 through 16 with tree nut allergy and is testing different immunotherapies to desensitize persons with tree nut allergy. This one's located in Hamilton. Also, you can go to clinicaltrials.gov to find research studies underway in Canada and in other parts of the world. In the search box that's pictured here, you can enter the condition, the study name, and Canada, for example, and you can select the study status to find out those that are currently recruiting. Current studies recruiting Canadians in Canada include BOOM, involving omalizumab with multi-food OIT. There's also one investigating peanut slit tablet that was talked about today. Both have multiple locations. There's also IMPROVES, which is a study investigating low doses of a food allergen and processed versions of that allergen for OIT, and TITAN, which aims to quantify food allergy thresholds with locations in Montreal and Hamilton, respectively. Following this session, you're going to get a short survey through GoToWebinar that asks your thoughts on today's webinar. And by the way, we did get a couple of questions today about the status of OIT in Ontario. We have recognized that this is a pressing issue in Ontario, and so we've been focusing some of our efforts in the province. And last month had an important discussion with the Ontario Ministry of Health and the Premier's office that have shown some openness to continuing the conversation and working towards a solution to better access on that treatment. So, thank you to everyone who helped advance our efforts there through your advocacy efforts and writing your MPPs and encouraging others to get involved. We will continue to update you on the progress via our newsletter as we have learned more from the government. So That's in that post-session survey. If you're there's a piece there that asks you if you're interested in doing some advocacy on access, please fill out the survey. And if you are interested in doing some advocacy work with us and that you haven't notified us in the past, please let us know. Also, there's many questions that came in about existing treatment. So here are 3 of the webinars that we would suggest you take a look at. One is, what's the big deal about Xolair? Ins and outs of clinical trials, goes a little bit more into detail in terms of the different clinical trials that are underway. And then there's one on food allergy treatment. What's food allergen immunotherapy and how is it advanced? So please take a look at that and visit foodallergycanada.ca/webinars to watch those. So lastly, Halloween is next week. We want all Canadians to shine a teal light on food allergy, help make Halloween a safer, more inclusive holiday for everyone. By shining a teal light, you're letting kids know that you have allergy-friendly or non-food treats at your door. And also take advantage of our tips and resources like the hacks and posters for your front door. Plus, don't miss out on getting your exclusive glow-in-the-dark bracelets. They're perfect to hand out to trick-or-treaters and to wear all year long to spark the conversation on food allergy. You can order them today to get them in time for Halloween. Visit foodallergycanada.ca/shine a teal light. And lastly, we'd like to thank our supporters. This webinar was supported by the Schroeder Foundation, the Peanut Bureau of Canada, and made possible from the donations of viewers like you. Thank you for your participation in today's webinar. You can view a recording of this webinar at foodallergycanada.ca shortly. Please also share it with others who you think may benefit from this content. This now concludes our webinar.